Welcome everyone to your first pre-lecture for Environmental 322, which is our environmental microbiology course. And this pre-lecture is going to be a quick review of hopefully some terms that you've seen in biology classes before. If you haven't taken bio, don't worry, we're going to cover the basic stuff in this lecture and we're going to cover the basic components as we go through the class. But if biology is something you haven't taken yet or maybe took a long time ago, ask questions in class so that we can make sure you understand the basic concepts. All right, let's dive in. So first things first, we're going to talk about microorganisms found in the environment. And generally when we talk about microorganisms, we're talking about organisms less than 100 micrometers in size. So where that red box is on this particular slide, that's what we're really talking about. We're talking about bacteria, we're talking about algae, protozoa, things that are small, can't be seen with the human eye. Now, size isn't the best definition because many times we have protozoa or algae or bacteria that are larger than this size, so it's not a hard and fast rule to say that something less than 10 micrometers is a microorganism. But in general, that's the practical definition that we're going to use in this course, so keep that in mind. When we talk about microorganisms, they can fall into three different categories. They can fall into the categories of bacteria, archaea, and eukarya. So multiple different ways of looking at things. Bacteria and archaea generally are considered singular cellular, but that's not always a hard and fast rule. But eukarya are generally considered multicellular. And again, not a hard and fast rule. Biology doesn't have a lot of rules because we keep finding new things. And what I want to point out here is that our prokaryotes are the bacteria and the archaea, and the eukaryotes are the eukarya, right? So we say we have two different classifications, prokaryotes and eukaryotes, and in this semester we're going to talk about both. We're primarily going to focus on prokaryotes in this semester with bacteria, but just be aware that eukaryotes are also really important. So within eukarya we have fungi, we have plants, we have animals, right? We have all sorts of different interesting things. Algae fall in there as well. So that's important to remember, is that when we talk about microorganisms, it doesn't just have to be bacteria, it doesn't just have to be prokaryotes, it can be all sorts of different things. So these are some ways that we classify life in this particular course, so just keep these in mind as we start to talk through things. I'm going to generalize mainly to microbes and bacteria in this course, but it doesn't mean that we can't have other microorganisms that are at play, so that's important, because other microorganisms have different structures, and we'll talk about structure a lot this semester in terms of how those structures matter to how we use technologies in environmental engineering. Another thing that I want to make sure that we briefly review is what we call the central dogma of molecular biology. And basically what this means is that DNA basically contains the information that encodes most life. Now, we do have RNA-based organisms, but in general, we start from DNA. And the central dogma of biology basically says that we have DNA, and something like DNA polymerase, which is an enzyme, will enable replication of DNA. But we also want to read the genetic code. So when we read the genetic code, we use something called RNA polymerase, which is another enzyme. And RNA works to help us translate what's in DNA into proteins and actual building blocks of life. So RNA works two ways. It helps to transfer information from DNA to proteins, and it does some actual work to create proteins as well. So you may remember from biology that there is multiple different forms of RNA. There's rRNA, which is ribosomal DNA. There's tRNA, which is transcriptase, transcribing RNA. And basically what happens is as that RNA reads the DNA, it produces proteins. And proteins are the workers in the cell. They make enzymes, structural components, they help with movement, they help with binding. Every organism needs proteins. And so in general, the central dogma of biology says that from DNA, we use RNA to translate those genetic pieces of information into workable pieces for the cell. And that's going to be important to us as environmental engineers because a lot of time understanding what types of proteins are at play is going to help us backtrack to what's going on in DNA and RNA expression. And that's a more advanced way of going after microbes in a system, but the idea that there is DNA, RNA, and proteins is important to understand. When we classify organisms, the classification that we showed earlier in these slides with these different branches, this is based on ribosomal RNA genes. Why wouldn't it be based on DNA? Maybe pause the video and ask yourself, why would we not base this tree of life on DNA? Did you take the pause? I hope so. So that's because not everyone has DNA, right? Not every organism has DNA, but every organism that we found so far has RNA. Now, I don't necessarily want to bring us into the debate about whether viruses fall into this category, but in general, everything that we've seen and we classify as life on Earth has RNA. And so if we're trying to classify organisms, it's better to do it with RNA instead of DNA, because not everyone has DNA. So just to kind of bring that home, right, we used to categorize organisms based on physical characteristics, but that doesn't necessarily tell us a lot about function. So RNA is the modern technique basically that we look at because it tells us what different protein structures are important. So we classify based on ribosomal RNA. So what's produced in the ribosomes in the cell. So if you remember from biology, your ribosomes are the place where we manufacture proteins. And so now, instead of looking at how similar are we by what we look like, we now look at what types of proteins we produce and what types of genetic material that we have. And so evolutionary relationships are now determined by our RNA. So that's kind of what some of these little figures on this screen are showing. They're showing different RNA sequences in different organisms. They're showing this, this is a ribosome down here with transcriptase and working through the production of proteins. And then what you see on the right side is kind of how we would organize by similarity different organisms. And you can kind of see that fungi are actually very closely related to humans, even though we don't look alike. But based on RNA, we're very close. Last thing that I want to mention here in terms of review is taxonomy. We're really not going to talk about the family and the genus and the order of all these different microorganisms. We're primarily going to see genus and species names. But just from a taxonomy standpoint, we start with all of life, and then organisms are organized into domains, then into kingdoms, phylums, classes, orders, families, genus, and species. So species is the most granular, right? So if you think Homo sapien, remember that the Homo component of that is the genus, the sapien is the species. So as you go from the top to the bottom on that little figure on the left of your slides, you're getting more and more granular when it comes to life. So hopefully you've heard genus and species before. Those are important concepts. Again, we're not going to worry too much about phylum and order and things like that. But if we know the phylum, the class, sometimes we know some characteristics about organisms, so it can be useful information to us. But we'll discuss that as we need it during the class. And that's just your quick review on some topics that hopefully you've seen in biology before, just a high-level quick chat about some of the things that I'm going to throw around when we're talking colloquially in class this semester.